Hello everyone, my name is Jie I'm a second-year PhD student at the University of Washington, Seattle. Thanks for being interested in our paper, Absolute 3D Pose Estimation and Length Measurement of Severely Deformed Fish from Monocular Videos in Real Fishing. This project is sponsored by NOAA.
I'm going to talk about these 4 parts one by one. First, its introduction. This is a video example from the dataset of this project. We can't see there is a fish, actually a Pacific halibut, is hooked on the fishing line and pulled up by a fisherman. This is called longline rail fishing, and our target fish is Pacific halibut. It's a kind of flatfish. The main motivation is to monitor the catching process and alert the fisherman when the halibut is shorter than the legal length. This is the backside of Pacific halibut. This is the belly side. We can't see Pacific halibut is really flat. So this is our task. Before measuring the fish length, we need to estimate absolute 3D fish pose. And the first challenge is that the fish is severely deformed, because it's bending during the catching process. So how to estimate the 3D pose for deformed fish is the first challenge. The second challenge is that the input data is monocular videos, which means we only use one single camera. So it's impossible to estimate absolute 3D information without additional input. But our proposed method overcomes these 2 challenges. To do this task, we use 2 off-the-shelf models in this project. One is YOLO detector. It models detection as a regression problem. It divides the image into an S by S grid, and for each grid cell it predicts P bounding boxes, confidence scores, and C-class probabilities. The other off-the-shelf model is called UNet, and it's for segmentation task. It models segmentation task as a per-pixel classification problem. We can't see it's an encoder-decoder architecture with skip connections. We also use chamfer distance as a loss in our method. So let me introduce it here. Given 2 point sets, chamfer distance is the sum of L2 distance to each point's nearest neighbor in the other set. For example, S1 is the blue point set, S2 is the orange point set. The first term in chamfer distance is those blue distances, which represents that for each blue point, we find the nearest orange point and calculate the distance. And the second term is vice versa, which represents that for each orange point we find the nearest blue point and calculate the distance. Next, I'm going to introduce the proposed method. This is the whole pipeline. In stage 1, there is the input image. Using YOLO detector, we can locate the fish in the image, though we crop out the bounding box, and use an encoder-decoder architecture similar to UNet, to get the mask of the fish. This mask is input to stage 2. Stage 2 is relative 3D pose estimation. Stage 3 is absolute 3D pose estimation and length measurement. Next I will introduce this deformable template in stage 2. To construct a Pacific halibut template, first, we download a binary mask of a standard flat Pacific halibut. Then using the foreground pixels, we construct the initial original 3D point set, which is the red point set in this figure. We denote the original point set as S0. Because halibut is a kind of flat fish, we set C values to 0, which means no thickness for simplicity. We define 4 deformable transformations to make the template deformable, which are scale, bending, translation, and rotation. There is 1 parameter for scale, 1 parameter for bending. The bending transformation is length preserving because we define bending as projecting the point set onto a cylinder, and the parameter R is the radius of this cylinder. The purple point set is an example after bending, so the length of purple fish is the same as the length of red fish. There are 2 parameters for translation, TZ equals 0 because we use orthographic projection. There are 3 parameters for rotation. Finally, after these 4 transformations, we denote the template's orthographic projection contour as S-template. We don't need all template points. Instead, we only need the contour points. For now, we have defined S-template. So in stage 2, given the mask of the target fish, we can define the chamfer distance between S-template and S-target, where S-target is the target's contour, obtained from the mask by using canny edge detector. Then we treat this chamfer distance as a loss and use gradient descent to minimize it, so that we can update all parameters in rotation, translation, bending, and scale. Here is an example about how to calculate the derivative. Here is a demo. From the input mask, we get the blue contour. The red points are the template. The green points are the orthographic projection contour of the template. The yellow edges and the light blue edges are the first term and second term in chamfer distance. We can't see those edges are like attractive force to make the green contour closer and closer to the blue target. We also predefine 3 key points in the template, which are head, center, and tail points. So stage 2 will output 3 key points in the image, which are these 3 black points. It also outputs 3 key points in relative 3D, which are these 3 black key points. Next, it's stage 3. As I mentioned before, because we only use one single camera, it's impossible to estimate absolute 3D pose without additional input. So in stage 3, using a checkerboard, we set up a world coordinate system shown in this figure. X, Y, and Z axis is the world coordinate system. We define Z equals 0 plane as our reference plane, and we assume when pulling up the fish hooked on the fishing line, the fish center is on this reference plane. So with this reference plane, and relative 3D pose from stage 2, we can do absolute 3D key points localization in the following way. This is image plane, and this bounding box and 3 key points in image. These 3 key points are actually these 3 black dots. This is camera coordinate system. This is the reference plane. Using camera intrinsic matrix, we can back project the center point onto this reference plane because our assumption is that the fish center is on this reference plane. So C' is the center key point in absolute 3D. Then we set up a temporary coordinate system at C', and this coordinate system is parallel to camera coordinate system. Then we can put the other 2 relative 3D key points in this temporary coordinate system. H' and T' are actually these 2 black dots from stage 2. Next, we can also back project H2D into 3D space using camera intrinsic matrix. And we know the absolute 3D head key point must be on line 1. And it is also must be on line H', C'. So the intersection point should be the absolute 3D head key point. We can do the same to T2D key point in order to find tail key point in absolute 3D. This localization method has a closed form solution. Finally, H', C', and T' are absolute 3D fish pose in camera coordinate system. The fish length is the length of arc H', C', T'. It equals the length of H', T' times a bending ratio. The below figure is an example. Here are 3 key points in image, like these 3 key points. This is camera coordinate system. Z is the depth. Z equals 0 plane is the camera plane. It is this image plane. These are 3 key points in absolute 3D, like H', C', and T' here. We can see this fish is about 5 meters away from the camera. Here is a demo for stage 3, because the initial bounding box are underwater, and the last few boxes are occluded. We only use green bounding boxes for length measurement. For each image, you have a measured length. Finally, we use a Gaussian distribution to remove some outliers and calculate the average length. Next is evaluation and ablation study. Due to the complexity of collecting data during longline fishing, we have 738 ground truth labeled fish lengths measurement histogram. We use 4 metrics: bias, EMD/RMSD, and KL divergence, to measure the difference between the ground truth histogram and the predicted histogram. For all metrics, lower value means better performance. Compared with the stereo method, our method is better in all metrics. About ablation study, we replace our chamfer distance-based optimization method with a brute force search method. We can see the performance is comparable. Also, for ablation study, we remove bending ratio and BFS and our method. The results show that both performance drop dramatically, which means bending ratio is very critical. The figure in the right shows histogram from different methods. Finally, its conclusion. We relax the requirement from stereo to monocular camera for fish length measurement task. We propose a relative deformable 3D SH template. We propose a chamfer distance-based optimization method to predict relative 3D pose only based on the 2D segmentation mask. Contour. We propose a 3D localization method with a closed-form solution to estimate absolute 3D SH pose, resulting in the frame-based absolute SH length measurement. From our experiments, we can see our method is better than a stereo-based method, which requires stereo image pairs as input and is harder to deploy in the challenging at-sea environment. Our optimization method has favorable performance as brute force search, which is more memory demanding and time consuming. And bending modeling is critical in fish length measurement. Thanks for watching.